Okay, so now officially welcome everyone. Very glad to see you all here in this new session of the BioClock Academy series. My name is Hannah Brooks, and I'm a PhD student within the BioClock Consortium, and together with Laura Pape, also a PhD student within the same consortium, we host these sessions. So the BioClock Consortium is a Dutch research organization that focuses on the biological clock and different aspects of it. So we research it in the context of healthcare, society, and in nature. And the specific aim of this lecture series is to provide you, but specifically young researchers, with the proper knowledge from experts in the field. So this series is hosted every third Wednesday of the month. For the timing of today, we will have about 14 minutes of talk, followed by roughly 15 minutes of discussion that will not be in the recording. You can put all your questions in the chat during the talk, so then we can come back to them later. You can also just ask them at the end. That's also fine. Yeah, I think that's the main thing. So then I leave the floor to Laura, who will introduce you to our speaker of today.
Yes, thanks, Hannah. Also, welcome from me, and it's a pleasure today that we have Dr. Selma Masri from the University of California, Irvine. And Dr. Masri is an associate professor of biological chemistry and is also affiliated with the Center of Epigenetics and Metabolism and the Chao Family Comprehensive Cancer Center. And her research focuses on how disruptions to our internal circadian clocks influence cancer development, and her lab explores how the circadian clock affects tumor growth in mouse models and how tumors can reprogram circadian metabolism throughout the body. They use genetic models, molecular biology, and large-scale omics tools to understand the complex interactions. And we are very excited to have you here with us today, and the stage is yours.
Excellent. Thank you so much, both Hannah and Laura. I appreciate the very kind introduction, also the opportunity to share some of our ongoing work. It's really a pleasure to be with you all today. I was asked to just do a very quick overview of my journey in science. And so my PhD training, I'm a cancer biologist, and my postdoctoral training, I did really focused on the circadian clock. And so when towards the end of my postdoc, I actually was entertaining ideas of both industry and academia in terms of career options. And I actually decided that I really wanted to be involved in not just driving, my own thoughts and ideas in terms of innovative research directions, but also to be involved in training the next generation of scientists. And I have to say that I consider this to be very, very important. I'm also incredibly proud. My lab, I have right now 7 PhD students. All of them are women. And I think that that's incredibly important because, I view training everybody, the next generation of scientists, especially women, to be, phenomenal scientists advancing in their careers and doing really well. And so this is something I'm incredibly proud of. Um, as Laura and Hannah mentioned, my lab is really focused on the intersection between how misalignment or disruption of circadian rhythms is important in driving cancer progression. And so let's go ahead and get into that. I'll be talking about 3 parts. And what I mentioned actually earlier is that what I'd like to do is give an overview of these 3 portions of my talk. And I would like to keep this a little bit more big picture, general bird's eye view to give you all a taste of what we do. I will spend some time going into some of the molecular details, but some maybe are not so important, and I will try to give a bigger picture overview. So for the first portion, I will discuss our work and how genetic clock disruption is involved in driving colon cancer progression. Part 2, I will be talking about circadian misalignment environmentally. And so we'll be talking about light pollution and, the artificial light at night exposure and erratic light exposure in driving carcinogenesis. And then for part three, we will focus on some tumor immunology. I understand immunology is not everybody's favorite topic sometimes. Again, I will try to keep this a little bit big picture. Okay. So I actually like to use this piece that was published in the New Yorker in 2007. It was a piece written by a staff writer at the time. His name was David Owen. And for any, it's a phenomenal piece for any of you that are interested in looking at it. And I just want to read one quote from this article. And what Owen writes is, for somebody standing on the north rim of the Grand Canyon on a moonless night, the brightest feature of the sky is not the Milky Way, but the glow of Las Vegas, 175 miles away. And for us as people who think about circadian biology and the importance of the biological pacemaker, we all know that this is incredibly important, and the issues of light pollution are global. This is an example from the United States. I'm sure many of you know where Las Vegas is approximately. But this is a major problem worldwide, and this was highlighted in Science in 2023. This was an issue focused on light pollution, and many articles that were featured in this issue of Science focused not just on the detrimental impacts of light pollution on our physiology as humans, but also on the animal kingdom and on the plant kingdom globally. And so this is it's a major problem. These are NASA composite images from the United States. I apologize. These can be viewed worldwide. I don't want to be United States centric in this talk, but I'm sure any of you can pick out your favorite major metropolitan area in the US. These problems exist globally. And so these are composite images taken at night. And I'm sure you all can really appreciate the major problem that light pollution has. So this is a piece that was actually a review article that was written by an incredibly talented graduate student who just defended her thesis last fall, Bridget Fortin. And I will be talking quite a bit about many of her different contributions in my lab. So this is a review article that is in press. It'll be out in about 5 days in Nature Cancer. And so Bridget highlights one, the molecular underpinnings of the circadian clock, but also two, the very important up and coming evidence in the field of carcinogenesis and the interplay with circadian biology. And so, of course, to this audience, the clock does not really need a major introduction. But as we know, the clock is a centrally controlled, light and trained pacemaker. The central clock in the suprachiasmatic nucleus then is responsible for dictating rhythms and synchronizing clocks in the periphery. Of course, we know that clocks exist in all of our tissues and every cell in our body, and this is really a genetically encoded transcriptional translational feedback loop, right? And so we have the positive drivers, clock and BMAL1, that drive circadian gene expression, including the expression of their own repressors, which then feed back to inhibit transcription. So for today's talk, I'm going to be focusing quite a bit on BMAL1 as we use the BMAL1 knockout to genetically disrupt or ablate circadian gene expression in a tissue-specific manner, and I'll be telling you about that. And the reason why we think the clock is very heavily involved in multiple aspects of cancer biology is because the clock touches on many, many different aspects of our physiology and cellular function. And so you can see, Bridget really beautifully highlighted multiple aspects that the clock touches on, including regulation of oncogenic signaling pathways. And I'll be talking about the Wnt beta-catenin signaling pathway in the gut today. I'll also be touching on some genome instability work that my lab is involved in, as well as changes in metabolism, stemness, which I won't be talking a great deal about today, but I will be focusing actually on the role of the clock in gating the anti-tumor immune response and regulating immunosuppression within the tumor microenvironment. Okay, so the the major observation that there has been, that there's a problem and a connection between circadian misalignment and tumor progression was first actually noticed many, many years ago in shift workers. And rotating shift workers, and this is based on a host of epidemiology data. And there's been a series of studies that have looked at rotating shift for many years. And these workers, these shift workers, actually had a higher incidence of certain cancer types. And this was actually summarized by the International Agency for Research on Cancer in collaboration with the World Health Organization, or the WHO, when they classified rotating shift work as a group 2A carcinogen. And the positive association between shift work and cancer was affiliated at that time with breast cancer, prostate cancer, and colorectal cancer. We do know now that the affiliation with these three cancer types has now been actually dramatically expanded, and work in many labs across the world has actually shown that both environmental as well as genetic perturbation of the clock is associated with many, many cancer types, including the three shown here, but also hepatocellular carcinoma, glioblastoma, blood cancers. And so really the list of pancreatic cancer is now really an emerging area. And so there's many, many cancer types that are associated with disruption, perturbation, or misalignment of the circadian clock. So for the purposes of today's talk, I'm actually going to be focusing on GI cancers, and in particular, colorectal cancer. And I like to actually show this data, and this is actually based on data from the National Cancer Institute of the NIH in the United States, but these trends are seen worldwide. This is not a US-based problem. So let's focus on colon cancer, which is shown here in the bottom portion. These are the rates of colorectal cancer in an older adults, 65 and older. And you can see these trend lines basically are on a decrease. This is excellent. This is because of pre-screenings and colonoscopies. And so this is very important because it really says that, early intervention really is an important mechanism to prevent cancer, right? And so this is excellent. This is very, very good news. The bad news, however, comes in this age group, and these are adults under the age of 50. So these are primarily adults in their 20s and 30s, and you can see that the trend line in these young adults is the exact opposite as it is in older adults. And this is quite a concerning trend line, and this has been on an upward trend for quite some time. And we don't understand the underlying etiology at all. There have been many thoughts and hypotheses proposed about what's happening. There are many potential putative risk factors: obesity, sedentary lifestyle, of course, diet. It's not just what you eat, but when you eat it. But also what's highlighted in this review in 2022 are sleep pattern changes and increased light at night. This has really been I think on the radar for many circadian biologists that, there could actually be an implication for the tie between early onset cancer and circadian misalignment in young adults. Again, these at this point are still major speculations. We need to really show this, but the idea that young adults have been raised with screens, artificial light at night, cell phones, all of this has been something that has been a major influence in development of these young adults. And so the idea is that there could be a potential link between rise in early onset cancers and the clock. So let's talk more about the cancer type that I'm going to tell you about today, which is colorectal cancer. So colon cancer or CRC is initiated by mutations in one very important tumor suppressor, and that tumor suppressor shown here, it's called APC. So approximately 80–85% of adult colon cancers have mutations in this very important tumor suppressor. This is important in driving the first initial initiating events of cancer. And in colon cancer, these are called adenomas. So one single hit, one point mutation in APC can drive a progression to an adenoma. However, this adenoma is not a cancer. This is a small polyp. This can be removed through colonoscopies, through screenings, and this is not really a full progression to cancer. What happens is that you have second drivers and second additional genomic insults. These mutations happen at very defined loci: KRAS, MYC, P53, SMAD4, PIK3CA. These second hits or second genomic insults are then responsible for driving progression. So the idea is you can initiate cancer by mutations in APC to develop an adenoma, and then you get second hits that are responsible for driving progression to a full-blown adenocarcinoma. So we simply ask the question, is the clock, and can the clock serve as a second hit? So does disruption of the clock drive the adenoma to an adenocarcinoma progression? So I mentioned APC. This is a very important tumor suppressor. But what does APC do? APC regulates a very important signaling pathway in the gut called the Wnt beta-catenin signaling pathway. This is not only important in regulating normal intestinal biology and intestinal stem cell function, but it's also a very key developmental pathway. The Wnt beta-catenin pathway needs to be controlled, really dynamically controlled. So mutations in APC would render this so-called, what's called the destruction complex, inactive. When you inactivate this destruction complex, this basically liberates beta-catenin. Beta-catenin is a coactivator. It can then freely move into the nucleus to activate transcription of TCF-LEF and drive expression of Wnt-dependent genes. So the idea is that high levels of beta-catenin, high levels of TCF-LEF and Wnt-dependent gene expression is very important. It drives oncogenic transformation and proliferation in the intestine. So really, Wnt high state is oncogenic. So we modeled this in many ways, and I'm going to be telling you in the first portion here about genetic circadian disruption and how this is important in driving colon cancer progression. And then in the second portion, I'm going to be telling you about circadian misalignment environmentally and how this is also important in driving progression to a full-blown adenocarcinoma state. So before I do that, I'm going to just briefly explain the mouse model that we use. So these mice, we actually crossed existing mouse models. So the BMAL floxed mouse was developed by Chuck Weitz's lab. And this is a floxed mouse under the control of Cree. And so this BMAL1 flox mouse, we crossed with an APC mutant mouse. This was developed by Tyler Jax's lab. Very important to point out here, this is a heterozygous APC mutation. So one mutant copy of APC, one wild type copy of APC. We crossed these two animals, and when we did that, we crossed that to get basically an APC heterozygous BMAL flox homozygous mouse. And this is under the control of villain Cree. So very important, villain Cree is expressed in the intestinal epithelium. So when we cross these animals with villain Cree, Cree is turned on in the intestinal epithelium exclusively, and this is really going to be driving the knockout of one copy of APC and both copies of BMAL1 in the intestinal epithelium only. And this is important because we don't really want to be disrupting the clock systemically. I'll show you more about that in the second portion of my talk. And so this was really important work driven by two postdoctoral fellows in my lab, Sungkook Chun, Rachel Fellows, and incredibly talented graduate student, Bridget Fortin. So what Sungkook, Bridget, and Rachel found, they crossed these animals. We isolated the intestinal epithelium, and you can isolate out the intestine, linearize the gut, roll it. These are paraffin embedded, so FFPE samples, H&E stained samples. You can see really beautiful intestinal staining. This is the normal morphology of an intestine in wild type here. The BMAL knockout on its own looks completely normal, and that's expected. Loss of the clock in the intestinal epithelium on its own without a second hit or stressor really does not do much in terms of overall histology and pathology. This is what the APC heterozygous mutation looks like. You can see these beautiful localized lesions shown here. These are adenomas. And these adenomas in two different animals shown here are blown up. And you can see these are nice local regions of small polyps. However, the problem really arises down here when we cross in the BMAL knockout. You can see in these boxed regions that are blown up, you can see that the BMAL knockout not only drives a complete disruption of the intestinal morphology, but it also drives local invasion into the muscle region of the intestine. And so this was graded by our pathologists to be a full-blown adenocarcinoma. So BMAL knockout is driving progression from the APC-driven adenoma stage to the adenocarcinoma stage. This was quantified here. In these slides, you'll always see that the double mutant, the APC-BMAL mouse, is always going to be in red. And you can see that versus the single APC mutation, we have an increase in tumor burden and a decrease in overall survival of these animals. Okay, so this is really bad news, right? Obviously, for these animals, you disrupt the clock and you're driving a progression, tumor progression in terms of CRC progression. But what's happening? And we use intestinal organoid cultures to dissect out mechanism. So the organoid cultures are really beautiful three-dimensional structures where we can take the intestinal stem cells, which are shown here, they're housed in the crypt. These intestinal stem cells that are shown in the turquoise down here can be isolated. We basically remove the villi of the intestine, we isolate the crypt regions, and we culture these stem cells with a cocktail factor of ENR, EGF, noggin, and R-spondin. These are growth factor cocktails that allow these stem cells to grow, divide, and proliferate. So this is based on the Hans Cleaver's method for intestinal organoid isolations. And so you can really beautifully see that we isolated these organoids from the intestine of all four genotypes. And when we started to culture these organoids, you can see that the double mutant, the APC-BMAL, really start to change in morphology. So the other three genotypes have what's called an enteroid structure. These enteroids are indicative of stem cells that grow, divide, and proliferate, and they look like little budding structures. That's because the stem cell is growing, dividing, and giving rise to itself and daughter cells that are differentiated cells. But what happened is that in the double mutant BMAL knockout, you can see actually that these organoids basically start to become cystic in structure. They are heavily proliferative, as shown by EDU incorporation. And these organoids basically start to grow in an unchecked, uncontrolled way. So this hyperproliferation phenotype and these cystic structures are indicative of tumor spheroids. They're also indicative of Wnt activation. So to dissect what's happening and to really understand mechanistically what's going on, we actually turned to a digital PCR strategy, and I'm going to save you guys a lot of work that went into discovering what exactly was going on here. We ended up identifying that clock disruption is driving loss of the second functional copy of APC. This is a mechanism in cancer called loss of heterozygosity. So as I mentioned, we started off with an APC heterozygous mouse. So this had one mutant copy and one wild type functional copy of APC. We identified actually after a series of experiments that clock disruption is driving genome instability, is driving loss of the second functional copy of APC. We isolated genomic DNA, and with the help of Nick Panunzio's group at UC Irvine, we developed a copy number assay for APC and identified that as we have these organoids transform from a enteroid to a spheroid form, they lose the copy of APC. We actually have 0 copies of APC left. This means that we have a gene conversion event whereby the wild type copy has now been converted to a mutant copy. In this case, we lose that second functional copy. We confirm this by whole exome sequencing, and the whole exome sequencing here is done. You can see the APC locus, which is shown down here. You can see the heterozygous reads, and the homozygous, in the homozygous mutant, you have that complete loss of APC. Now, what does APC LOH do, or loss of heterozygosity? When we lose APC completely, as I mentioned, this is part of the destruction complex. Once we lose APC, we hyperactivate Wnt. Now, this is very important because Wnt signaling, as I mentioned, is heavily oncogenic. It drives not only a proliferative program, it also drives a glycolytic program. And I'm not going to get into the details of this. So this is actually activation of c-Myc and activation of glycolytic metabolism that we trace by C13-based metabolomics to trace glycolytic metabolism. But just trust me to say that we have an activation of growth, proliferation, glycolytic metabolism in these organoids as a consequence of BMAL1 loss. Okay, now that's all in mouse models, and we do actually quite a bit of human work in my lab. We have a hospital located not far from our main campus, and our surgeons actually collect normal tumor tissue as well as tumors from patients that are going in for tumor resections. We get this human colon material in the lab, and we derived patient-derived organoids in the lab. We infect these patient-derived organoids, or PDOs, with a BMAL1 luciferase reporter to really understand what's happening in normal versus tumor epithelium. And you can see beautifully, this is one patient, you can see these beautiful oscillations in BMAL1-driven luciferase activity, really indicating that human colon has a beautiful rhythm in circadian gene expression. These are dexamethasone synchronized. And what is really striking is that in the tumor, we completely lose that rhythm. And this was also recapitulated in big human data from the Cancer Genome Atlas, looking at several hundred colon cancer patients versus normal tissue. Okay, so to summarize the first portion of my talk, I showed you all that genetic clock disruption drives genome instability, loss of heterozygosity. This leads to Wnt hyperactivation and Wnt signaling that drives a proliferation and a glycolytic metabolic phenotype. But you know, whenever I give this talk, the idea here is that this is a mouse model, and as humans, we don't walk around with a BMAL1 loss in our intestine, right? That just isn't that is not a normal thing. And so we use the genetic model really to model what's happening in humans. But what is happening in humans is the artificial light exposure and the circadian misalignment that we know is happening worldwide. And, you know, we are not the first to think about this. I cited actually several very important papers in the field looking at circadian misalignment models in many, many cancer types that I mentioned at the beginning of my talk. And I think that we've really, as circadian biologists, we've really started to make the case that, you know, we need to really be thinking about circadian misalignment in the context of cancer. So let's get into that data. We model circadian misalignment in 2 ways in my lab. One is through extended light, and the other is through circadian misalignment through shift or chronic jet lag. So let's talk about the extended light first. We take mice, and you know, as we all know, mice are housed on a 12-hour light, 12-hour dark paradigm. We extend the light to 16 hours of light, 8 hours of dark, or 20 hours of light and 4 hours of dark. And this is just the locomotor activity in metabolic cages to confirm that. But what happened in these APC mutant mice, as we extend the light, you can see that the tumor progression is getting much more severe. And this is shown in the light paradigm shown here. As we extend light, we see a decrease in survival and an increase in tumor burden. So this is really striking, right? This is an APC mutant mouse only, and we're changing and extending the light paradigm, and this is actually driving cancer progression, right? I mean, as circadian biologists, for us, this is incredibly striking. And I think we need to really be sounding the alarm on this data. We modeled also rotating shift work. And so we do this by phase advancing the light cycle every 2 days. This models shift work 3 to 4 days a week. And we called this mouse model a shift disruption, or SD paradigm. You can see that the shift disruption completely disrupts locomotor activity in these animals. And when we take the APC mutant mouse and we subject it to shift disruption, or SD, you can see that this also in parallel drives tumor burden, intestinal tumor, not just burden, but also intestinal tumor size, and it also decreases survival, which I'm not showing here. So this is incredibly important and incredibly striking data. But what's happening and why is that? We've started to dissect mechanism. We have isolated these organoids and subjected them to sequencing. We see that there is a genetic insult at the APC locus, but we also have seen that there's genome instability everywhere. And so we've mapped SNPs, so point mutations, insertions and deletions in collaboration with Nick Pananzio's lab. And we're really seeing that circadian misalignment is driving genome-wide instability, mutations genome-wide, and these hits, we believe, are very important in driving cancer progression. So what does this lead to? It leads to an intestinal phenotypic plasticity. And, in the interest of time, I'm not going to really spend a huge amount of time on this, but the genome instability is leading to hyperactivation of Wnt, Notch, hedgehog, many very important pathways in the intestinal epithelium, which are driving the phenotypic plasticity and stemness in the gut, which we think is an important driver of cancer progression. So just to summarize, I briefly touched on this data. So environmental circadian misalignment really phenocopies what we see in our genetic model, in that when you environmentally perturb the circadian clock by just changing the light, we're driving changes to the genome and insults to the genome, where not just at the APC locus, but genome-wide, we're seeing instability and mutations caused by circadian misalignment. This is driving Wnt hyperactivation and phenotypic plasticity that we think is an important driver for colorectal cancer. So, I think this is incredibly important, and it's really arguing the fact that, we really need to be thinking about cancer as a very important insult, and a potential hit really could be the circadian clock. Now, we really need to show that this is important for those trending upward cases in the young population that could really be impacting young onset colon cancer. So in the last short period of time, in the last maybe about 10 minutes or so, I'd like to touch on this really beautiful work that was carried out by Bridget Fortin in my lab. And Bridget is an incredibly talented student. She just graduated and defended, and she's in industry now working on tumor immunology. And what is really important about Bridget's work is that she came to my lab with a major interest on anti-tumor immunity. And as I said, I know, for some people, immunology is not everybody's favorite thing. I'm going to try and keep this a little bit more high-level immunology. But what Bridget did is she isolated immune cells from the intestine, from these genetic mice that I mentioned in the first portion of my talk, the BMAL knockout, the APC, and the double hit mutant, so the APC BMAL, where we have very serious cancer progression. And this is based on incredibly important work in the circadian field that's really been showing a very important crosstalk between the clock, cancer progression, and tumor immunity. So Bridget took a single cell sequencing approach. And this is some of her data that she took and looked at. And just in the interest of time, I'd just like to call your attention to the clock disrupted state, which is shown here. And let's look at neutrophils. These are a very important immune activating first response cell type in the intestine. And so you can see in the BMAL knockout shown in green, there's an increase, a small but significant increase in the BMAL in the neutrophil population in the BMAL knockout. This is dramatically expanded once you initiate cancer in the APC adenoma state. And this is even further driven in the adenocarcinoma, the double hit model. And Bridget really quantified this, and this is shown here by flow cytometry. So Bridget took these myeloid cells, so these are what are called neutrophils, monocytes, and macrophages, and she quantified them by flow based on very specific and defined cell surface markers, which I won't get into, but if anyone has questions, I'd be happy to answer that. So you can see in the BMAL knockout state, so without a tumor, in the BMAL knockout state, only clock disruption, loss of the clock in the gut is able to activate the myeloid cell compartment. And strikingly, very, very interestingly, it suppresses a very important cell type in the intestine, which are called cytotoxic CD8 positive T cells. These are cells in the body that are very important immune responders. They are going to be recognizing tumor cells or any cell that is not self, and they're going to be killing that, targeting and killing that cell type. So you can see that the clock is suppressing that cytotoxic T cell population. This is even further exaggerated in the cancer setting where we look at the APC versus the APC BMAL. Again, same trends, increase in myeloid and a decrease in the cytotoxic T cell proportions. Now, what's really striking about this is that this is really suggesting that the clock is suppressing anti-tumor immunity. These cells, the cytotoxic T cells, are very important responders in terms of the anti-tumor immune response. They're going to be the first defense mechanism to target the tumor. How is this potential mechanism happening? So let's first jump into this. This is how it works. T cells express what's called PD-1. And this is a very, very important cell surface marker that's a receptor for its ligand PD-L1. The T cell is going to be recognizing its ligand expressed on the tumor cell. This is going to be telling that T cell, this is a non-self cell and we need to be targeting and killing this cell. What happens in the case of cancer progression, when you're promoting cancer, you have an activation of immune cells that can interfere with this recognition. What that means is that you have myeloid cells that can begin to express this ligand here, PD-L1. This will interfere with this recognition here and will interfere with the ability for T cells to recognize and kill tumor cells. So the neutrophils that we identified in our data set, interestingly, express by our single cell sequencing PD-L1. They recognize the ligand that is recognized by the T cell, by that receptor expressed on T cells, meaning that these myeloid cells could potentially interfere with the recognition mechanism of T cells to recognize tumor cells. So this very important immune checkpoint, we measured and we really assayed for activity. And again, in the interest of time, I don't want to spend a huge amount of time on this. Bridget quantified this and really showed that in both the clock disrupted state and the tumor clock disrupted state, we have an activation of these immune cell types that are PD-L1 positive, meaning these cells are potentially, quote, immunosuppressive. These are myeloid cells that can suppress the anti-tumor immune response, and we quantified these cells that are so-called myeloid-derived suppressor cells, or MDSCs. These myeloid cells we co-cultured with T cells. This is a gold standard assay to actually show that they're immunosuppressive. And when we co-culture them with T cells, you can see that they beautifully suppress T cell activity and proliferation. So clock disruption is an important mechanism to prime a cell type that can interfere with T cell recognition of tumor cells, right? This is very, very important. But, I've been telling you that the clock is an important mechanism and gating strategy for recognizing immune cells. And what was very interesting, Bridget actually did a really cool experiment where she profiled these so-called immunosuppressive myeloid cells at 2 times a day. In the inactive state, shown here, and in the state of activity, which is shown here. And you can see that the state of activity in these animals have a much, much higher level of these immunosuppressive cells than in the inactive stage. And so what Bridget thought and what we were discussing at the time, can we leverage this? Can we basically create a mechanism by which we can target and show increased efficacy for delivery of therapies at the optimal time of day? And this is a concept that's been raised by many in the field, especially Francis Levy, and I'll get into this work towards the end of my talk. But the idea is ZT16 in the active phase of the animal is when these suppressive cells are high. If we treat with anti-PD-L1 immune checkpoint blockade at this time, which is when these cells peak, can we see an increase in the efficacy of this drug in terms of targeting and keeping the cancer in check? And so that's exactly what Bridget did. She treated at ZT4 and at ZT16. This is when these myeloid cells that express PD-L1, when hit with anti-PD-L1, she hit them with the immunotherapy at this time point. And this is actually exactly when she sees the best response in terms of shrinking of tumor size. And she went on to show that this is not just important in colon cancer, which is shown in our subcutaneous models here. So MC38 is a colon cancer cell line, but she also did this with lung cancer, shown here, as well as melanoma. And these are two cancer types that really beautifully respond to immune checkpoint blockade or immunotherapy. And she always sees that in comparison to the active or the inactive phase, the active phase always seems to have a better response to immune checkpoint therapy. So this is very, very important. It's showing, so Bridget's work really showed that immunotherapy can be leveraged in terms of efficacy at certain time of day. We profiled this based on when those immune suppressive cell types peak. And this is because of this rhythm in those cell types, this circadian rhythm. And this is an important concept. The idea that delivery of drugs can really be timed based on chronomedicine or chronotherapeutic approach has been championed by many in the field, especially, as I mentioned, Francis Levy, who's been looking at chemotherapy. But these are actually retrospective clinical studies that have now been looking at timing of immunotherapy and looking at the survival of patients. And what's really striking about these retrospective trials is that these trials have shown that based on timing of delivery, the beginning of the active phase of humans, we are seeing a better response to immunotherapy in certain cancer types. These were in melanoma and non-small cell lung cancer. So this has really launched a series of clinical studies worldwide. And this is the summary of our work in the news and views that was highlighted by Nature Immunology as well as Nature Cancer, really showing that our work showed that clock disruption is important in promoting an inflammatory response. It promotes a very specific immune cell type, and these immunosuppressive immune cell types have a rhythm. The rhythm based on time of day, we leveraged this and we actually showed that treating with immunotherapy based on when these cells peak, when their levels are highest, this is actually when we can see the best outcome in terms of colon cancer, lung cancer, as well as melanoma. And so this is incredibly important. A big thank you to my group. I get to go everywhere and talk about this work, but really they're the ones really doing all of this work. This is my lab in its current state. This was a recent picture we took. I have the privilege of mentoring 2 incredible postdoctoral fellows and 7 PhD students. And they are an amazing, amazing team to work with. I've also highlighted their funding sources, which I think is very important. They're all incredibly successful in their own right in terms of the work that they're doing, the grants that they're securing. And I'm incredibly thankful to work with such a talented team every day. A big thank you to many of our collaborators, Nick Panunzio, Kai Kessenbrock, and Devin Lawson, for their collaboration on the single cell work, Francesco Marangoni, our amazing collaborator in terms of immunity, anti-tumor immunity, and many, many others that have helped us along the way. Our funding sources, of course. And I'm a couple minutes over time, but I think we still have time, plenty of time for questions. Thank you again for the invitation. This was really a pleasure, and I look forward to answering your questions. Thank you so much for your attention.